All right, let's go over question 6 of homework 4, and then I'll go over each of the answers. There you go, that, yeah. Okay, so I explained to you this pathway here, or what these things mean, and I'm going to go over the answers. So the whatever I marked with a check mark, that means that those are true, and then these unchecked are not true. Let's see why. So the first one, the wild type big A, so that's that one, the big A DNA, encodes a transcription factor that induces gene expression of the anthocyanin pathway. That is true because that's what I said up here. I told you that the A gene encodes a transcription factor, and that's the one that is involved in making a pigment. So somewhere up here, I was telling you about making the pigment. So that big A allele is the code for the big A mRNA, and these are all introns that get spliced out. And then this intron here is a large intron that in the big A allele gets spliced out. In the little A allele, just part of it gets spliced out, and that's what makes a little A protein. So this little A protein is a transcription factor that ultimately will lead to white flowers, and the big A transcription factor will lead to the formation of pigment. So when it says it's a transcription factor, it's true in both of them, right? But we know that it induces the enzymes in the anthocyanin pathway. So the anthocyanin pathway is a pathway for making color. So if it induces, you get color. If it represses, you get absence of color. And that's why the first one is true and the second one is not true. Because if the big A inhibited making color, then this would be the white one and this would be the purple one. Okay, so this is an example of a positive regulator. Okay, the third one, the little A allele encodes a white pigment. That one is not true. So you would think, okay, the little A is making white pigment, but what the little A encodes is a transcription factor that is shortened, and because it's different from that, then it doesn't work in inducing the transcription of the genes that are responsible for making pigment. So it's not that it encodes a white pigment, because this would mean that it encodes maybe an enzyme that will make things white, that were colorful and then they are white. Instead, it doesn't encode an enzyme that makes a white pigment. It encodes a transcription factor that will promote transcription of a gene that will make things purple. But whenever the transcription factor doesn't work, then you don't make pigment. So I guess the key things here is, the main thing here is that it encodes a white pigment, but it doesn't encode a pigment. It encodes transcription factors that will lead to the flower being white because of lack of the transcription of the enzymes needed for formation of pigment. So let's see, the first one, although the spliced mRNA of the little A allele is longer than the big A allele, the protein is shorter. That one is true. So the big A allele removes this many nucleotides in that intron. The little A allele removes this many fewer. You can count. Okay, well, 8 nucleotides. So instead of removing a whole 5,775, it removes that minus 8. So it removes fewer. So the mRNA for that is longer, is 8 nucleotides longer. But the protein is shorter, see here, because the stop of translation is the star. So and you realize that from seeing, oh, the protein shorter, so this is a stop codon that got put here. And they're like, but if the same sequence is here, how come it's not a stop codon? Well, because there's a frame shift. Maybe that's what I'll ask you in the exam. Because there's a frame shift mutation here. So when you put 8 nucleotides, the frame changes, the reading frame changes, because 8 is now multiple of 3. And then when your reading frame changes, then you could potentially get early stop codons. So that one is true. The mRNA is longer by 8 nucleotides, but the protein is shorter by however many amino acids were excluded here because of the early stop codon. Okay, then the next one, P flower is haplosufficient. You know that that's true because the heterozygote. So here, this, let's see, in here you cross purple with white, and he got 100% purple. So you know that these guys are heterozygotes. And since the heterozygotes look like one of the parents and not somewhere in between, then you know it's complete dominance, which I told you the complete dominance happens because of haplosufficiency. So even though the heterozygote has this shorter transcription factor, this transcription factor doesn't influence the action of the longer transcription factor. So the haplosufficiency is this transcription factor does the job even if you have only one copy of it, and this second copy doesn't mess with the action of that one. If it influenced the action of that, then you would have something that is somewhere in the middle. So that would be incomplete dominance. But you know, it's complete dominance, so it's haplosufficiency. And then the last one, considering data P for the flowering locus, if the little a allele was disrupted the action of that, then you'd expect a heterozygote to be white. So if it fully disrupted, that is true. So let's see how would that happen. So this is a transcription factor that will act on transcribing, let's say, some enzyme that makes this pigment. So this is some gene that will affect the transcription of another gene, and that gene makes things colorful. And then this one is some transcription factor that does not allow transcription of those genes for making color, therefore is white. If this transcription factor was hogging the promoter that this dysfunctional transcription factor wants to bind, then that would be like somebody standing in the kitchen, like you were the one cooking, and this one is your toddler or whatever. It's that person or that person that doesn't know what they're doing. If they sit on the side and they're not interrupting you, you can make the dish that you were trying to make. But if they're competing for space or pots or whatever, then you cannot get to this state. So if the action of this transcription factor affected the action of this other transcription factor, then you wouldn't get the dominant color. You would get white if it fully disrupts the option on that, or somewhere in between, which should be like you have some pigment made, but not all pigment made. So perhaps I have to say here, if it fully disrupted the action. If you didn't mark this because you're like, well, maybe white, maybe pink, then you're thinking right. But maybe I would just say it would not be like the dominant phenotype or the dominant allele.